This video covers false prediction number 7. Evolutionists say that they can tell how long a species has been evolving by looking at its genes. Is that true? Let's have a look. Linus Pauling was a great American scientist in the 20th century. He successfully predicted protein secondary structure, not so much with DNA structure. He incorrectly predicted a triple helix instead of the double helix. He also thought that the nucleotides were pointed outwards rather than inwards. So not so successful on that prediction. He made one other famous prediction, which is the topic of our false prediction number 7 in this video, and that is the molecular clock, along with Emil Zuckerkandl in a couple of papers in the 1960s. So what is this molecular clock? So here is a notional evolutionary tree. You have some extant species there at the top. A couple of them in the yellow circles there have a common ancestor in the blue dot. Let's zoom in here. So one of the questions that evolutionists ask is, well, how much time has elapsed since the speciation event? So you have these two species. Let's zoom in here. So you have these two species that have been evolving since they're split off from the common ancestor. How long has that been? So let's just take an example. Let's say one is a cow and one is a horse. It's got a common ancestor. They have a common ancestor. How long has it been? So obviously you can look at fossils, but you don't always have fossils available for all species, and fossils aren't all that accurate. So another approach that Pauling and Zuckerkandl looked at was using genetics. So let's look at a protein here, cytochrome C. Cytochrome C is a common, well-studied protein. It is found in the cow and the horse, and therefore, if you're an evolutionist, you're going to believe that it's derived from the common ancestor. So you have this cytochrome C. Now, when you look at the proteins that are a sequence of amino acids, you compare the cytochrome C in the cow and the horse, they're not exactly the same. In both cases, it's a cytochrome C. It's got the similar structure, similar fold, similar function. But the sequence of amino acids is highly similar, but not quite the same. There's some differences. So the idea that Pauling and Zuckerkandl advanced here is those differences that you see in genes, such as the cytochrome C, accumulated since the common ancestor. By definition, if you're an evolutionist, the common ancestor had the common ancestral version of the cytochrome C, and it's been evolving ever since in these two independent lineages. The more differences you find, the longer it's been since the common ancestor. So you know mutations occur at some roughly constant rate. So you can back out how long it's been just by looking at the differences in the cytochrome C. And so here's a typical plot you'll see of the cytochrome C differences. So each one of these dots is a comparison, the number of differences between two species. So there, I'm circling the human and the cow, for example, and the number of differences is about 75 in the gene, and that is a lot more than, say, the horse and the donkey, which only has about, say, 10 differences. So that makes sense on an evolutionary perspective. Horse and the donkey are very similar. Their common ancestor is relatively recent. They shouldn't have that many changes. Whereas the human and the cow, a lot more time since their common ancestor. They've been evolving separately for much longer, should have more differences. So, kind of makes sense. And you'll see that the dots kind of fall into a line here. The horse and the cow example, kind of in the middle there. About 50 or so nucleotide differences. So the idea is you can use the differences in the genes as a sort of clock. In the case of the horse and the cow, you're looking at something like, 65, 70 million years since the common ancestor. So it's a molecular clock. Very clever. This was well received by many evolutionists. In this case, here's a quote: "The discovery of the molecular clock stands out as the most significant result of research in molecular evolution." So quite quite a quote there approving of the molecular clock idea. Here's another one. One of the simplest and most powerful concepts in the field of evolution. Another quote from Ernst Mayr. Zuckerkandl and Pauling showed that many, perhaps most, molecules have a rather constant rate of change over time. Such molecules can serve as a molecular clock. So it became an icon of evolution, if you will. And here's an interview of Zuckerkandl, and the author of this paper and interview refers to what they found in the 1960s there as a discovery, the discovery of the molecular clock. So it's a real thing. There really is this thing, this molecular clock thing, and it was discovered by Zuckerkandl and Pauling. Okay, so the prediction is there is a molecular clock for past speciation events. That's the prediction. This is false prediction number 7, because we're going to find out that this is a false prediction. And let's look at the falsification. First of all, it was found that different protein genes must evolve at different rates. So you don't have a universal rate of differences building up in these proteins. You see here in this plot, the cytochrome C that I just showed is shown in red. Hemoglobin is in blue. The fibrinopeptides is in brown. They have different slopes, so they build up these changes at different rates. So the clocks run at different rates, put it that way. And evolutionists can explain this. Here's an explanation from a blog. They tick at different rates for different proteins as shown below. This is because the sequences of some proteins, like cytochrome C, are highly constrained by natural selection, i.e. they're conserved. So you can't change all of the amino acids. Some of them are necessary. So only the ones that can freely change will change, will reflect those changes. Other proteins like fibrinopeptides can tolerate many more changes. Okay, so that's the explanation for evolutionists. Now, the key phrase there is it's highly constrained by natural selection. It's an explanation. However, what the author doesn't let on, or what evolutionists don't normally tell you, is that there's no evidence for that. That is an inference. That is an assumption to save the theory. Evolution requires that because otherwise it's not going to make any sense. So that's not observed, that's inferred. There's no evidence for that. In fact, we've looked at this. This is assumed to save evolution. Let's take a look at this plot again, but zoom in. What that earlier plot did not show was histone 4, a protein we've looked at before. Histone 4 is way down near the horizontal axis there. It hardly changes at all, so it's highly constrained. So in the case of histone 4, evolutionists would have to say that the clock ticks very slowly because you can barely change it at all. Now, we looked at that in false prediction number 6, recall. That was histone proteins cannot tolerate much change, and that's false. So it's not backed up. Let me back up here again. Histone 4. This is assumed to save evolution, but what we saw with false prediction number 6 was experimentalists have changed the histone 4 and experimented around with that, mutated it, and it doesn't seem to hurt it very much, and it still works pretty well. So it's not clear that histone 4 is in fact highly constrained. It appears that it is not, and so the whole story kind of falls apart. So let's move on to the next problem, the next reason why this prediction was falsified. Same protein but different lineages must evolve at different rates. So remember, just a minute ago we looked at different proteins have different rates. The clock ticks at a different speed, so to speak. In this case, the same protein but different places in the evolutionary tree, different lineages must evolve at different rates. If you're an evolutionist, for this to make sense, you have to say, we've got more variation in the results here. That's the data are not fitting the molecular clock idea very well, but I can try and fix that by saying for some reason the clock ticks at a different speed in different lineages. So now we're starting to add on more epicycles, more patches to the theory because the data are not cooperating. And here's a paper to this effect. Different types of proteins diverge at vastly different rates. Moreover, the same type of protein has been observed to evolve with different rates in different phylogenetic lineages. And you'll notice that phrase here, has been observed to evolve. Again, that's false. It has not been observed to evolve. That's inferred, not observed. Skipping down, strong and lineage-specific regional effects were seen on the molecular clock rate of protein divergence. So here we're now seeing regional effects, but again, I want to point out were seen. No, they were not seen. These effects, these variations in the clock were not seen. They are inferred. What's seen is extant species with extant proteins and genes, and the comparisons between them. This is all inferred. Okay, so summarizing the failures so far, the molecular clock is not universal with respect to protein type, taxonomic lineage, and geographic region. Moving on, protein genes must evolve at different rates over time. So now we're seeing a temporal factor. Depending on what epoch you're looking at, you might have different rates the molecular clock might click at. Again, trying to fit the data. The data are not cooperating with the theory. Here's a paper, Estimating the Rate of Evolution of the Rate of Molecular Evolution. Clever title there. Point being, yeah, it's changing over time for some reason. So another problem. The molecular clock is not universal with respect to time now. Another problem is that it is contradicted by the fossil evidence. We talked about that earlier. The fossil evidence can provide dating, and they don't fit the molecular clock data. They don't align. Large discrepancies have been found in dates of evolutionary events obtained using the molecular clock. Twofold differences have been reported between the dates estimated from molecular data and those from the fossil record. Here's another example of that. This paper used fossil paleontological data and tried to use that to calibrate the molecular clock, but they got conflicting results. The global molecular clock was clearly violated. So another problem here, another failure. Molecular clock is contradicted by fossil evidence. Next, different molecular methods are inconsistent. Furthermore, different molecular methods can give dates that differ 20-fold. This research implies that many previous studies, even some of those using recently developed methods, might have placed too much confidence in their date estimates, and their conclusions might need to be revised, indeed. So, more failures here. The molecular clock is internally inconsistent. Next, the observed mutation rate versus the inferred mutation rate don't align. There's a mismatch. So reading from this paper, studies of molecular evolutionary rates have yielded a wide range of rate estimates for various genes and taxa. Recent studies based on population level and pedigree data, this is something you can, more like a laboratory measurement of mutation rate, have produced remarkably high estimates of mutation rate, which strongly contrast with substitution rates inferred in phylogenetic studies. So the inferred mutation rate, if evolution is true, mutation rates must be this, is much lower than what we actually measure. Our results show that it is invalid to extrapolate molecular rates of change across different evolutionary timescales. So another failure here, the molecular clock is contradicted by observed mutation rates. Different proteins contradict each other in interesting ways, and this paper highlights that. Two proteins that were seen to contradict each other are glycerol 3-phosphate dehydrogenase, GPDH, and superoxide dismutase, SOD. This paper looked more closely at that. It also added in a third protein, xanthine dehydrogenase, XDH, to try and kind of solve the riddles, see if XDH could solve the problem, and maybe it could identify the outlier. If XDH fell in line with GPDH, then superoxide dismutase would be the problem, for example. So bring in a referee here, a third protein. Well, they just got more confusion. Let's read this. It has been observed that the variance of the rate of evolution is generally larger than expected according to the neutrality theory, which has raised the question of how reliable the molecular clock is, or indeed whether there is a molecular clock at all. Skipping down, we have carried out an extensive investigation of 3 proteins, glycerol 3-phosphate dehydrogenase, superoxide dismutase, and xanthine dehydrogenase. We have observed that, first, the 3 proteins evolve erratically through time and across lineages, and 2, the erratic patterns of acceleration and deceleration differ from locus to locus, so that one locus may evolve faster in one than another lineage, whereas the opposite may be the case for another locus. It's all over the map here. The observations are inconsistent with the predictions made by various subsidiary hypotheses proposed to account for the over-dispersion of the molecular clock. In other words, there have been set forth some subsidiary hypotheses to the molecular clock to try and explain these problems. They brought in XDH, they looked more closely, and it's just not working. Skipping down here, the hypothesis of the molecular clock of evolution emerged from early observations that the number of amino acid replacements in a given protein appeared to change linearly with time. So we looked at that earlier with cytochrome C, for example. Indeed, if proteins and genes evolve at constant rates, they could serve as molecular clocks for timing evolutionary events and reconstructing the evolutionary history of extant species. And they give us a reference here. There's the classic Zuckerkandl and Pauling paper. Skipping down, we have observed that 1, rates of evolution change erratically between and within lineages, and that 2, the patterns of change for different genes are discordant. Skipping down, the GPDH rate of evolution is much slower between species of Drosophila than between species of mammals, whereas the rate is much faster in Drosophila than in mammals for XDH. So you have these different proteins and their genes contradicting each other. Skipping down here, the extent to which the erratic patterns of evolution of GPDH and SOD are an aberration rather than representative of prevailing modes of protein evolution was left in abeyance in the earlier work. We now show that XDH also evolves erratically and following an evolutionary path that is different from those of either GPDH or SOD. This just isn't working. Skipping down, our observations for all 3 genes are inconsistent, not only with the molecular clock predicted by the neutrality theory, but also with subsidiary hypotheses, such as what we've looked at here, that predict that patterns of evolution will be consistent across genes, even if variable between lineages for any given gene. Continuing, we are left with hypotheses that attribute rate variation to the vagaries of natural selection, whether as a consequence of functional shifts that change rates of neutral mutation through time differently for different genes in different organisms, or in response to phenotypic evolution and environmental heterogeneities. These 2 hypotheses amount to a denial of there being a molecular clock. So it just isn't working at all. When you resort to natural selection, I want to point that out in that first sentence there, we're left with the hypotheses that attribute rate variation to the vagaries of natural selection. The whole idea of the molecular clock is based on the neutral theory of evolution. You have to have the neutral theory to some extent working. When you have to resort to natural selection, you're giving up. You're throwing out the molecular clock, the fundamental assumption behind the molecular clock. And so you can see here they're kind of, knocking over their king here. They're giving up here, capitulating. So another failure here, the molecular clock is contradicted by observed mutation rates. We talked about it earlier, but now on some specific examples of specific proteins that contradict each other. Overall, we have a falsification of the molecular clock, as pointed out in this paper, which reads, numerous follow-up studies, including ours, have falsified the concept of a universal molecular clock. Work on fossil sequences also invalidated the molecular clock. The molecular clock is now considered to only exist in limited situations. Okay, so we have seen the prediction. We've seen a representative papers on the reasons why it is falsified. What about the outcome? What about the response? What has happened since then? How do evolutionists reckon with this? Well, first of all, they're going to make their models more complicated. They're not going to give up on the molecular clock. It's an icon of evolution. You could still find it in contemporary papers. But obviously the models have to become much more complicated. There is a lot more moving parts, and there's a loss of parsimony, therefore. This is the typical pattern in evolutionary theory. You have simple beginnings, and it just gets more and more complicated because the science contradicts the theory. The empirical data contradict the theory, so you have to have epicycles upon epicycles. As this paper reads, most workers now agree that a global molecular clock does not exist, and that rates can change between lineages and within a lineage over time. New methods of estimating branch length do not assume a strict clock, but the number of models for molecular evolution is then effectively infinite. So they're making the point, once you start to float these variables and you allow the clock to click at a different rate, to tick along at different rates for different proteins, at different time points, at different time epochs, in different lineages, and so forth and so on, it's just a tautology, and you can just tweak the knobs all day long to make the model work. And you're violating parsimony. Now this paper is going to quote some other works here. Buckley admitted that, quote, molecular clocks are notoriously difficult to apply because of many well-recognized problems, the most difficult of which is that different lineages evolve at different rates. Skipping down, Neer and Sanderson noted that, quote, with respect to rate heterogeneity, once the model of molecular evolution departs from a simple one-rate molecular clock, the divergence time problem enters a realm of model selection in which the number of models is effectively infinite. So making the same point here, once you start to parameterize everything and allow things to float, you're just violating parsimony and you're adding epicycles upon epicycles. Now, the next problem is more fundamental. Once you start looking at the molecular clock and uncovering all these problems, it starts to reveal something a little more fundamental, quite a bit more fundamental. And that is the loss of underlying fundamental evolutionary model. Let's read on here, see what I mean here. The idea of a strict molecular clock is less popular now than it was for a few years of the last decade. However, roughly clock-like evolution, in which evolution proceeds more or less continuously, although not at exactly the same rate, in different lineages, has been a common assumption in biology since the acceptance of evolution. It is seen in the almost ubiquitous idea that greater divergence between taxa generally implies a longer time of evolution. So in other words, this general idea of the molecular clock is just so fundamental to evolutionary thought, it long predates of the molecular clock idea. if you just look at a morphological level. And so they're going to quote here some different people here. Even this rough clock makes the quite unwarranted assumption of more or less equal rates of evolution in different groups. And so going back to 1872, Hutton concluded that, quote, differentiation of form, even in closely allied species, is evidently a very fallacious guide in judging of lapse of time. So in other words, he's pointing out this isn't working. It doesn't fit the evolutionary model. This fundamental evolutionary thinking that informs the molecular clock idea is just not working. Crozat later made similar observations. He criticized the universal clock approach and made the opposite assumption, namely that different groups in the same region can and do usually show tremendously different rates. Continuing, he also argued that rates can change dramatically within a group. So this is just an interesting sidelight here, that it goes beyond just falsifying the molecular clock. It really gets at something more fundamental. The species data do not fit the evolutionary tree. It's phylogenetically problematic. So that's what these problems with the molecular clock are revealing. Another problem here is the loss of the neutral theory. I talked about that a minute ago. That is so fundamental to the molecular clock assumption. The neutral theory of evolution has to be working to some extent. It doesn't have to dominate, but you have to have some level of the neutral theory working. And it's basically what we're finding with the problems with the molecular clock is how much, to the significant extent to which it doesn't work. And you lose the neutral theory. And yet the neutral theory is so ubiquitous and pervasive in the literature. Let's read from this paper here. The conclusion is that molecular evolution is dependent on the fickle process of natural selection. Natural selection is over against the neutral theory. Loss of reasonableness, another problem. This paper reads, the erroneous inferences that could be reached by assuming a molecular clock for any of the three genes investigated are dramatically illustrated in Table 2. If we use the average rate of evolution of Drosophila over the last 60 million years for estimating the time of divergence for other organisms, the divergence of the three multicellular kingdoms is estimated at 7,000 million years by GPDH, a gross overestimate, yes indeed, but at 450 million years and 398 million years, which are blatant underestimates by SOD and XDH, respectively. Similarly, erroneous and disparate divergence times would be inferred for other groups of organisms. Indeed. Okay, next, loss of the fundamental premise of clock-like rates. This paper reads, since Emil Zuckerkandl and Linus Pauling proposed their hypothesis of a molecular evolutionary clock in their seminal 1965 paper, biologists have been fascinated by the prospect of adding a temporal dimension to their inferences of evolutionary relationships of genes and organisms. This variation in evolutionary rate, however, can cause severe problems in molecular evolution dating and the inference of phylogenetic relationships. Phylogenetic methods implement evolutionary models in which each branch has an independent rate of molecular evolution. While these models revolutionize phylogenetic inference, they cannot disentangle evolutionary rate and time. So you have this problem of, okay, now you're going to float all these variables and allow for different rates and different lineages. So was the rate faster or was the speciation event sooner? It's difficult to disentangle these things. But in a landmark 2006 study, Drummond and colleagues developed a Bayesian Markov chain Monte Carlo method to co-estimate phylogenies and divergence times, allowing each branch in a phylogeny to have a different evolutionary rate, co-estimating substitution and relaxed clock parameters. So you just get more and more complicated, and you can fool yourself into thinking that you're solving the problem. The relaxed phylogenetics approach also enabled measuring the clock-likeness of genes, revealing that clock-like evolution is relatively rare and that most genes exhibit non-clock-like rates of sequence evolution. So what I was getting at a minute ago here was the loss of parsimony. This particular model that they're talking about in this paper is just more complicated, more parameters to fit the data, which are not cooperating with the model. But they realistically and honestly reflect on this result that, okay, once you've got this more complicated model with more parameters being adjusted, you also can reflect back on each of the genes and determine, are there any of them that actually are have a constant rate of evolution? And they find, well, no, actually. So remember, that was the fundamental assumption behind this whole molecular clock enterprise. In addition to the neutrality, that the neutral theory must be alive and well to some extent, you also have to have this clock-like behavior of genes. At least a particular gene must have some clock-like behavior, even if it's not consistent with other genes, even if it's not consistent with other lineages and so forth. But what they're finding here is, well, yeah, it just doesn't really fit. So the whole theory is just falling apart here. You have a multiplicity of explanatory mechanisms, as you might expect. With all of these problems, there's all kinds of explanatory mechanisms. And I'll read to you a few of these examples. Determining the relationships among and divergence times for the major eukaryotic lineages remains one of the most important and controversial outstanding problems in evolutionary biology. Aha. Skipping down, yet relationships among these groups remain poorly understood because of saturation of sequence changes on the billion-year timescale, possible rapid radiations of major lineages, phylogenetic artifacts, and endosymbiotic or lateral gene transfer among eukaryotes. Well, there you have it. A list of the usual suspects comes out. They have this cadre of explanatory factors that can explain just about anything. And you have, of course, the saturation, but look at this, possible rapid radiations of major lineages. Anything can happen. It's evolution. Phylogenetic artifacts. There's all kinds of artifacts that just happen. And of course, lateral gene transfer. That can explain all kinds of things. Reading on, estimating the divergence dates between the major eukaryote lineages using molecular analyses is even more difficult than phylogenetic estimation. Yes. Error in such analyses comes from a myriad of sources, including calibration fossil dates. I think that means, I should say, calibrating fossil dates. The assumed phylogenetic tree, the nucleotide or amino acid substitution model, substitution number estimates, the model of how rates of evolution change over the tree, error inherent in the time estimates for a given model, and how multiple gene data are treated. Again, all kinds of explanatory factors for the errors that we see. By reanalyzing data sets from recently published molecular clock studies, we show that when errors from these various sources are properly accounted for, the confidence intervals on inferred dates can be very large indeed. Okay, so here's another illustration of this multiplicity of explanatory mechanisms. Molecular dating has now been adopted in many areas of biology. This broad uptake has been due partly to the development of Bayesian methods that allow complex aspects of molecular evolution, such as variation in rates of change across lineages, to be taken into account. But in order to do this, Bayesian dating methods rely on a range of assumptions about the evolutionary process. which vary in their degree of biological realism and empirical support. This is classic. So you have a range of assumptions. Well, yes, indeed. There's a lot of parameters that are floating, a lot of epicycles upon epicycles. So yes, there is a lot of knobs here that are being adjusted. But I love this, that vary in their degree of biological realism. In other words, some of them are not biologically realistic, and they also vary in their degree of empirical support. In other words, some of them are not empirically supported. This is wonderful. These assumptions can have substantial impacts on the estimates produced by molecular dating analyses, indeed. Here's another example of the multiplicity of explanatory mechanisms. For over half a century, it has been known that the rate of morphological evolution appears to vary with the time frame of measurement. Rates of microevolutionary change measured between successive generations were found to be far higher than rates of macroevolutionary change inferred from the fossil record. Yeah, so there's that problem we talked about. More recently, it has been suggested that rates of molecular evolution are also time-dependent, with the estimated rate depending on the time scale of measurement. So yeah, again, we talked about that. This followed surprising observations that estimates of mutation rates obtained in studies of pedigrees and laboratory mutation accumulation lines exceeded long-term substitution rates by an order of magnitude or more. So we talked about that also. Although a range of studies have provided evidence for such a pattern, the hypothesis remains relatively contentious. Skipping down, we review the various biological and methodological factors that can cause rates to be time-dependent, including the effects of natural selection, calibration errors, model misspecification, and other artifacts. So again, we have this list of the usual suspects. Explanatory factors, explanatory mechanisms are always available. This has important consequences for the use of molecular clock methods to estimate timescales of recent evolutionary events, indeed. Okay, so we've seen the prediction, we've seen the falsification, and now we've seen the response, the outcome, the denouement. How did evolutionists respond to that? And it wasn't pretty. What does this say about the theory? In particular, we're looking at these theory virtues, and how does it impact these theory virtues? Well, they failed, and they failed badly. We're looking at evidential accuracy, causal adequacy, internal consistency, internal coherence, universal coherence, simplicity, durability, and fruitfulness. And I must say, it really does fail badly on all of these. I don't think I need to go through these, except on the fruitfulness might be the one that you might question. Because there are a lot of papers, and you probably could just see that from the few that I cited here that I read from. Over the recent decades, there's been many investigations, many studies, many papers written on the molecular clock and associated ideas. And so you could say, well, there's some fruitfulness there. Well, I don't think so, because as you could see, what the studies were looking at were they trying to save the theory. The studies did not reveal new information, new discoveries. The failure of this prediction did not lead to new knowledge, did not lead to, this is it, this is why it failed. Now we don't understand. Instead, what we see is a broad attempt, an ongoing broad attempt to save the theory and trying to figure out, well, how can we fix this? And it's just leading to more and more loss of parsimony, more complexity, and not a resolution. So you can hardly call this a fruitful. It's just more of the same. It's just getting worse. So I think it fails on fruitfulness as well. I can't, you just can't give this a break on fruitfulness. So religion drives science, and it matters.